So it's a pleasure to introduce Dr. Alberto Pugliese, a friend and a colleague. He's a worldwide known researcher in the area of type 1 diabetes. He recently retired from University of Miami as of October 2022, where he was the Enlo and Eugenia Professor of Medicine and Microbiology and Immunology, and Deputy Director at the Diabetes Research Institute that some of you had the opportunity to visit yesterday, to took a new position as a professor and chair of a new department of Diabetes and Immunology at the City of Hope in California. And he will talk today about team science approaches and the value of early screening.
Thank you, Francesco. Good morning and pleasure to be here. And I'll cover the topic that was assigned to me about team science and early screening in type 1 diabetes. As a disclosure, I have served as consultant for Prevention Bio. And I will start by saying that in the past 35 years that I've been involved in type 1 diabetes research, I think I have witnessed tremendous progress. And I think that's an important message to communicate to our patients, family members of patients, which often are frustrated by perceived actually lack of progress, which is not completely true. What is true is that the progress in science takes a long time to translate into actual change that patients and families can appreciate. But I think many of you have seen how much diabetes care has changed and also are aware of how much diabetes research, even clinical research, has been changing and improving because of these discoveries. And discoveries that have been made include much improved understanding of the pathogenesis of type 1 diabetes and the natural history of the disease, the identification of target autoantigens of this autoimmune disease, the identification of genetic factors that not only control the risk of developing the disease, but also modulate its progression as people are advancing from a preclinical stage towards clinical diagnosis and beyond. We have been able to improve the prediction of type 1 diabetes, identify novel biomarkers, some of which are in the process of being validated. And we have been moving progressively from secondary prevention, which traditionally is when you already have signs of autoimmunity triggered in a patient, towards primary prevention when children especially may have a genetic predisposition but not necessarily have activated the autoimmune disease. Trial organizations have been conducting a variety of clinical trials, and now we have, at least for some treatments, we're able to see therapeutic effects in terms of either delaying diabetes development in prevention or after diagnosis, maintaining insulin secretion for a period of time. And you're probably all aware of the advances in transplantation, whether it's pancreas or islet, and more recently stem cell-derived beta cell replacement strategies, which are now in clinical trials. And we also have sophisticated disease models, mouse models, or in vitro models that really allow us to understand the mechanism of disease much better. And then, of course, the technologies for improved insulin delivery, insulin therapy, and glucose monitoring, and so much more. But what's important is that a lot of this progress really would not have been possible if we had not been able to organize ourselves as communities of researchers and apply team science and collaboration essentially towards these goals. These are all achievements that in isolation is honestly not possible to achieve. So I'm going to cover some of the major organizations that have been active in the field. And these include, and this is not a completely exhaustive list, but just a few. The Type 1 Diabetes Genetic Consortium that focus on discovering genes associated with the disease, the Type 1 Diabetes TrialNet, that is a clinical trial organization primarily in the United States, but also in Europe and Australia. The Immune Tolerance Network, which is another clinical trial organization, although not absolutely specific for Type 1 diabetes. The TEDdy study, or the Environmental Determinants of Diabetes in the Young, which is a clinical follow-up from birth, trying to identify especially environmental factors that are important in Type 1 diabetes. And similarly, the Diabetes Prevention and Prevention Study, DIP, in Finland, And another organization, which is also a clinical trial organization in Europe called Inodia, which I'll describe briefly. And the Human Island Research Network, which has started about a few years ago, 5 to 6 years ago, that it's more of a basic type of organization, very much focused on studying beta cells, and as well as the autoimmune process in a variety of settings, but focusing very much on the human system. And then the Network for Pancreatic Organ Donor with Diabetes, or MPOD, which I'm a co-director of, that focuses on the study of pancreas and other tissues from organ donors with diabetes. And that's, I think, a prime example of team science as well. And so if we look at the genes, for example, over many years, the table is really hard to read, but it basically says that the consortium collected genetic material, DNA samples, and conducted extensive genotyping on families with affected sibling pairs with type 1 diabetes from many regions of the world. And so that was a very exhaustive process and comprehensive to try to understand the genetic basis of the disease and take into account genetic heterogeneity and in different background populations. And at the end of the day, the graph shows the odds ratio of any particular gene or locus to give you increased risk of diabetes. And very clearly, the HLA region gives you the highest risk, and by many times higher than any other genes that was identified later. And then you can see that there is over 50 genes now that have been linked to type 1 diabetes risk. And that's important because, number one, it tells you that there are many genetic factors involved, but this information allows us to actually understand what genetic pathways, what molecules could be actually therapeutic targets that we could try to address in the clinical trial. So, for example, the IL-2RA, which is the IL-2 receptor A chain gene, it's a very important risk factor, and now is being tested in clinical trials as a therapy, and so forth. So there are many examples of how this information is actually very, very valuable in terms of clinical translation. Now, and many of these genes are not just expressed in immune cells being autoimmune disease, but many of them are actually expressed in the beta cells themselves. So that tells you that the beta cells is also subject to genetic regulation of the disease process. And again, this is important for therapeutic consideration. The NPOD, or the Network for Pancreatic Organ Donor with Diabetes, basically was funded in 2007 upon the recognition that if we really want to understand the disease and the pathogenesis, we really need to look at the disease tissue, at the pancreas with diabetes. It's not possible to do this just looking at experimental mice or rodents because, number one, although the disease can be very similar, there are factors that are unique to human. And so one example is exposure to environmental factors. You're familiar with the idea that, for example, viruses might play a role in human type 1 diabetes, but in mice, they live in a sterile environment. So they're not exposed to the same viruses that we are. So it's very hard to model those infections, for example, in a mouse. And so, or if you're trying to understand what are the sequences of the T cell receptors that are involved with type 1 diabetes, you can study them in the mouse, but they have no relevance to human. So the only way to do it is to study human. So that was recognized, and what we did was we launched an effort working with organ procurement organizations. And since inception in 2007, we have now recovered close to over 500 donors in different categories, but close to 180 or so donors with type 1 diabetes. Many of them actually have a fairly short duration of disease, where it is much more likely that you are going to see signs of active disease in the pancreas, including persistence of some beta cells, but as well the inflammation, the autoimmune disease that leads to beta cell loss. And also, we basically started a general population screening by screening for autoantibodies, which are risk markers for type 1 diabetes, the organ donor population. And with that program, which is really something difficult to do, we have been screening thousands of donors over the 15 years, and we have identified about 50 or so that actually express autoantibodies associated with type 1 diabetes. And this is very important because it allows us to identify donors where we may actually see earlier signs of the diabetes pathogenesis and whether it's environmental factors or alterations of beta cells or immune abnormalities, we can study those. And what's important is that it's not those three people in the picture that do these studies. It's actually the scientific community. And I spend a lot of my time in this effort actually bringing people together and promoting team science and creating working group and getting people together to collaborate. And, you know, for example, the virus question, I started a working group that was active for several years. And the idea was, okay, everybody wants to look at the virus, but everybody has one assay they use. How about if we put it all together, we integrate that effort? And so now we have data from multiple assays looking at RNA, proteins, or whatever we want. And we have a much comprehensive picture of the situation. So that concept has been repeated over and over and over. And essentially, we have created a community where people can interact within the consortium and even outside the consortium and trying to find collaborators and essentially try to advance the research as fast as possible. And I always tell people, we owe it to our patients, we owe it to our organ donors. Every time that you study a tissue from a donor, you're talking about someone who lost their life. And sometimes that loss of life is accidental, could have been a car accident or anything else. But sometimes, way too often, when we're talking about children with diabetes, sometimes they pass away because of diabetes, whether it's a severe hypoglycemia or consequence of DKA. And so that is still happening, not frequently, fortunately, but it's still happening. And we want that to stop. Now, so we have scientists all over the world, and this is our website. If you're curious, I encourage you to visit it. It's www.jdrfmpod.org, and you'll find a lot of information about this research that has been done and who's involved and what. And this is examples of actually loss of pancreatic beta cells and insulitis, or the inflammation of the islets. You see a couple of islets there. The brown color are immune cells attacking the islets. The red color is the glucagon on the left panel, or my left. And in the upper right panel, where it says insulin Ki-67, the pinkish color is insulin. You can see that in a normal islet, that would have been all red, and it's not. So there was already beta cell loss in organ donors that did not have diabetes, but tested positive for autoantibodies for type 1 diabetes. And these donors are quite rare, and unfortunately, quite often they actually get transplanted. And we have fights with the surgeons because they let it go to research. You're so rare, it's not going to impact your present, but we publish this, but still, we lose many to transplant. For many, many studies from many investigators, I put this picture together, trying to imagine what we have learned about the pathology, the evolution of the pathology, the disease over time, considering different stages of the disease development. So at the single autoantibody positivity level, which you're probably familiar with the idea that it's the initial stage sort of autoimmunity, but at that stage, your risk of diabetes is pretty low. We don't see beta cell loss in the pancreas, we don't see insulitis, we don't see increased expression of HLA class 1 molecules, which is a feature of the pathology of type 1 diabetes in islet cells. And these features begin really to become known when you have multiple autoantibodies, which goes hand in hand with the increased risk that individuals with multiple autoantibodies have. And in clinical classifications are called stage 1 type 1 diabetes. And if they have dysglycemia, which in organ donors we cannot determine, they would have stage 2 type 1 diabetes, and they would be considered having more than 75% risk of diabetes in 5 years. And then you have the time of onset where, again, you see that these phenomena become more and more prevalent. But then over time, these continue even years beyond diagnosis. So the process is not really over a diagnosis. It continues for a number of years, about 10 years. And after that, really, most of what is left is beta cell loss. This is the most profound. So, but there is really a much wider time window, I think, for intervention and for studying than previously anticipated. The TEDdy study I mentioned before is concentrated on environmental factors, but has done a lot more than that. I just want to mention it. And in terms of environmental, they actually have demonstrated, and their data go hand in hand with the data that we generated looking at the pancreas in input donors, that it appears that there are chronic infections, either sustained over time or repeated recurrent infections that are associated with increased risk of type 1 diabetes. So essentially, we don't find evidence for acute infections that massively attack the beta cells, but just few cells that are positive and attacked by viruses, but that triggers inflammation. So, and to have a parallel between a clinical study and the organ donors is very, very important. And so the crosstalk between these consortia is very important to continue. This consortium in Europe, INODIA, is really a clinical trial organization and as well a study that trying to understand the disease mechanism by following patients over time and even before clinical diagnosis, and has put together a group of scientists that are more like basic in nature and basic science types, where a lot of different studies can be done. And it's a highly organized organization. What I want to highlight of this organization is it's actually a bit unique because not only is funded by the European government or grants from the European government, but is also supported by industry. So it's a partnership. And the idea is that this partnership from the outset can actually facilitate the translation of discoveries and clinical trials and then into new therapies. And so I think that this is a very visionary approach for this type of initiatives. And the other thing is that in this consortium, you do have a very strong participation of patients and their families, even in terms of deciding what type of things or research should be pursued. So that's something that perhaps resonates with this audience, where I believe there is also an interest in patient participation and representation. And these are actually clinical trials that they are pursuing at the moment. And see, they have 4 clinical trials that they have in the plans. And I don't know that it matters to describe each one of them, but these are some therapies that are actually quite innovative, and some of them have never been tested. The ATG trial, MELT ATG, is actually that's been tested before, you'll see in a minute, but this is trying to determine the lowest possible dose. And then the impact study is using an immatope, which is really an industry-derived molecule that is antigen-specific. And so this is very, very innovative. And verapamil is based on a trial that has been done recently showing preservation of C-peptide. And then the last one is a humanized anti-CD40 monoclonal antibody that blocks co-stimulation. So there is a combination of newer, highly innovative, and already tested, but trying to advance further down towards translation. So quite interesting approach. TrialNet is based largely in the United States. It's supported by NIDDK, the National Institute of Health. And the goals is to delay, prevent, or modify the course of type 1 diabetes. And they have instituted history study. They basically screen relatives of patients with type 1 diabetes for autoantibodies. They follow them over time. And on occasion, they have prevention trials that they can do and then randomize the patients there. Or they've been running also new onset trials, whereas upon diagnosis, people are randomized into an intervention or a placebo group, trying to identify therapies that actually either delay diabetes or preserve insulin secretion. And at the same time, again, promoting team science approaches and collaboration, they've been bringing together a lot of scientists and trying to analyze the samples that are obtained from all of these subjects and either understand the natural history of the progression of the disease, and I'll show you some examples, but also trying to understand how these therapies work, which is very, very important. And there are sites in the US, but also there have been sites in Europe and in Australia. And it started in 2000, so it's a 20-year plus organization. These are examples of results from some clinical trials that have been done by TrialNet. A couple of them involve the anti-CD3 monoclonal antibody. And as you can see, the blue lines in these graphs represent treated patients, and the red are placebo. And what is shown is the insulin secretion, residual insulin secretion over time. And so you can see that in the treated groups, in all of these trials, there is a better preservation of insulin secretion in the treated group as opposed to placebo. But also you can see that sometimes there is a response that almost stabilizes insulin secretion, but then once you stop treating, it continues to go down. Or in some cases, for example, the abatacept study, you can see that it keeps declining. We never really stopped the decline, but it is less than the placebo group. So there is a lot to learn, but we are learning. And in this particular trial, which was a low-dose thymoglobulin treatment, alone or together with GCSF or placebo, and the important line is the either blue or is the blue line, the ATG only. You can see that afforded a better preservation of C-peptide. And this is quite dramatic because if you look at compare from baseline to 1 year, the treated group at 26% decline versus 58% in placebo. And at 2 years, between 1 and 2 years was only 23% in the treated group versus 38% in placebo. And overall, from baseline to 2 years was 43 versus 74. So this is still not perfect. You're still losing C-peptide, but about half of if you were not treated. And the treatment here is actually 2 days of therapy. That's it. So there is a long-lasting effect. And now we're hoping to launch next year a trial where we will combine this treatment with low-dose IL-2 for a year and see if we can stabilize and prevent further loss. So, but that results is 5 years from now. Now, a lot of lessons have been learned from TrialNet cohorts. An example here, putting together all the information that came from the genetics and applying now in the clinical setting. And so with this group of individuals, we developed sort of a genetic risk score, which was combined with a metabolic score that Jason Sanker from the University of Miami had actually defined and validated. And you can see I'm putting out a dead arrow, which points out at those two groups that are quite differentiated. And it really allows us to, combining those two, to tell those people apart in terms of progression. And that's very important because it helps you stratify people for risk for certification into a clinical trial, whether it's based on the aggressive nature of the therapy or time, for example. And so time to diabetes is a very important concept, and I'll bring it back in a moment. These are studies that we did. We were trying to discover some new biomarkers for risk, and we focus on circulating microRNAs, that are small microRNA molecules that the levels may vary. And in this study, what we saw is that we could identify levels of certain microRNAs, which ones are, it really doesn't matter, that actually conferred increased risk of progression to diabetes among young children, relatives, or patients who had multiple autoantibodies. So technically, the belief has been once you have multiple autoantibodies, it doesn't matter, there is not much more that you can do to improve risk prediction. But what that tells you is actually is that we were able to increase the odds ratio by several fold by including microRNAs in this assessment. So this is something that we're in the process, we would like to validate. And because, you start with, a cohort and then you need to reproduce in other cohorts, and hopefully some of these things will translate into better prediction for risk. Just recently, a combination of cohorts from TEDI and TrialNet showed that even in the normal range, an increase of 10% in the A1C value is actually associated with increased progression to diabetes. Again, that's very important. Actually, a very easy parameter to measure, and it can be followed, and it allows you to really improve the prediction. If you look at those curves, they're very, very different. Here, these are studies from Jason Sanker from TrialNet, and those very funky curves, what they're basically trying to tell you is that when people are beginning their progression towards type 1 diabetes, there is actually an increase in insulin secretion, which is basically shown in those graphs. But once you reach an inflection point where you go backwards and your insulin secretion starts declining. So there is a compensation at the beginning and then further loss. And that transition, for example, could be used to say, okay, now things are serious. You're going to get closer to diagnosis. So that's all important because it helps us understand how the disease progresses at the level of the beta cell. And also we can use that information for patient stratification. This is a very beautiful scheme that Colin Diane put together a couple of years ago, and really allows you to visualize how it is important that we think about risk, but the highest risk, the people with the higher risk that we would want to treat the most because they're the one closest to diabetes. They really represent 1 in 10,000 of the general population. So think about that for a moment. If we had to find all of those, we really need to have collaboration and wonderful, powerful screening machineries to identify those individuals and randomize them in the trial. So just a simple concept of how difficult this has been. And but the screening is happening and is highly valuable. This is actually a quite recent publication where they talk all about screening. I invite you to go and take a look at it. This is the slightly revised version of the stages of type 1 diabetes, which finally, to my pleasure, they put a stage 4, which is established type 1 diabetes. But bottom line is there is a lot of screening going on. And if you look at these tables, we have type 1 diabetes screenings that screen relatives for clinical trial participation. Example on TrialNet, as I mentioned, the Inodia, the Bar-Oxford study in the UK, and the type 1 diabetes screening in Australia. And then there are a lot of general population screenings. That's very important. We have general population screenings now that are growing. And so that table at the bottom actually starts there and it continues over 2 other slides. And I'm not going to spend a lot of time on it, but we're now screening the general population, essentially newborns. You can use a combination of genetic and autoantibody screening, which actually helps. And then there are all kinds of things you can do, whether it's treatment, randomization to treatment, or just follow-up. And but this is very important that it's happening because it's where we're heading, right? And that's where preventative strategies are going to be in tests. And also, the value of early screening, this is one study, the most recent one. There are other studies before that show that if you screen for risk of type 1 diabetes, you can actually help reduce the occurrence of DKA and help preserve insulin secretion and diagnosis. And that's very important practical implications. Again, DKA can be very, very dangerous. If you can decrease the risk of DKA, it's incredible value for a subject. This is the results of really putting into work the whole of it, right? The ability to identify that 1% of people that have the highest risk of type 1 diabetes and testing therapies in that. And so this was done by TrialNet. This is the anti-CD3 antibody. And as you can see, they were treated, and it was actually a very cleverly designed study because they were looking at time to diabetes development. So they could do a study in a really short period of time with a small number of subjects, compared to more traditional study designs where you would need 300 people and follow them for 7 years before you can call the trial. So that had very important progress also in terms of trial design. And basically what happens is there was a much reduced development of diabetes is 43% in the treated participant versus 72% in the placebo group. And there was a reduction in the odds ratio of type 1 diabetes. And basically a significant delay. And this is actually very important. We're modifying the disease course. Now, is it the end? This is the final solution? No, it's not. But we can build on it. And so we need to add on therapies that can continue to protect the beta cells in the long run, but they need to be safe. And they need to possibly be given chronically because the disease is chronic. Post-diagnosis and pre-diagnosis, the figure on the left shows TrialNet studies, is C-peptide decline or insulin decline. And you can see that it varies by age group, but importantly, the decline becomes much more rapid about a year, 6 months to a year prior to diagnosis, and it continues more aggressively for the first six months. So that's important because prior to diagnosis, you actually have sort of a sluggish progression. But then at some point, the disease gets into gear and then it accelerates. And we don't know exactly why that is happening. But if we could figure that out, it would be important. And if you put it side by side with pancreas pathology study from EMPOD, you can see that even donors with multiple autoantibodies, it's very hard to measure a quite significant decline in terms of loss of beta cells. The red bar is the proportion of beta cells in the islets, and it's pretty much like normal. But in a recent onset, that's where you see a very drastic reduction in beta cells. And then with long duration, they're all gone. So these two things are actually consistent. The other important information is that when you look at diagnosis, within 1 year from diagnosis, 88% of patients in trial subjects maintain a stimulated peak C-peptide greater than 0.2 picomoles per ml. That's the entry threshold for participation in clinical trials. And so, but when you look at 2 years, it's 66%. So what's important here that typically new onset trials are done within 3 months from diagnosis. But if at 1 year you have 88% meeting that standard, we should still keep enrolling beyond 3 months. And then we would have many more patients in trial because within 3 months enrolling in a trial, many people just miss the opportunity, just not enough time. And I think we're giving up too early on the patients. So I'm glad that this data exists. And I think you'll see an opening up of the criteria towards later, longer duration of disease in coming years. This is another study that we did in terms of microRNAs. We just measured microRNAs in the circulation on the same day that the near diagnosis received the mixed meal tolerance test to evaluate residual insulin secretion. And selected microRNAs, the levels of those basically divided these patients into groups. Those that had higher residual insulin secretion and those that had lower residual insulin secretion. If you look at the curves, and I don't know if I don't see a marker, but if you look at the upper right panel, that's peptide insulin secretion. And so you see that there is a red line and the blue line, right? So the red line is higher and they remain higher for a year. So basically with one measurement at near the time of diagnosis, we could predict the decline of insulin secretion for up to a year. It's actually longer, but we stopped the analysis in 1 year because we had more subjects. And then those curves at the bottom, what they try to depict is glucose versus insulin secretion. And so, and what it tells you is that those in the red, which had better C-peptide, yes, in fact, maintain better C-peptide. You see the red line that is more to the right, and it's actually lower. That's because the glucose is lower, which is on the y-axis, and the insulin secretion is on the x-axis. If it is to the right, there is more insulin being made, right? And over time, you have the baseline, the 6 months follow-up, and the 1 year follow-up. You see that the red line is moving up and towards the left, which it's indicating progression. But when you compare that red line over time to the blue line, which is the people identified by the microRNAs as having lower insulin secretion at onset, you can see how much worse those guys are doing. So basically, with a simple measurement, you can actually predict the future course. And again, that can be helpful in terms of strategizing and randomizing people to trials. So I hope that I gave you some examples of team science efforts, but they need to all be integrated, and they really are advancing type 1 diabetes research on multiple levels. We need to do better and do more, and this will continue to require very well-organized team science approaches, but also partnership with the biomedical industry, which really is needed to bring things to fruition, and collaboration between private funding organizations and government agencies, and of course, the participation from patients and family members, without which we can do anything. These collaborations and flow of information needs to occur within consortia and across consortia. And with these studies, we're now really in a better position to identify risks very early in the process, even from birth. And we're going to be able to test therapies that are a lot less toxic than the therapies that we use later in the disease course, where we try to manipulate the immune system. Maybe we can, manipulate the microbiome or vaccinate or things of that nature that are not necessarily bad therapies or aggressive therapies. And I've shown you an example of how early screening actually has even clinical benefit and multiple stages of disease. So these are all important lessons that we're learning. And again, none of this would have been happening without team science approaches. And there are tricks that one can do to make team science work. I can talk for an hour about that. There are actually people that have written papers about team science. I discovered that several years ago after I started putting together working groups and implementing team science. I said, oh, it has a name. I realized that it had a name. And then I invited this guy to give a talk, and this guy gave a talk, and I was going, he was saying, to have successful team science, you should do this, you should do this, you should do that. And I was going check, check, check. I'm doing all of these things. Nobody taught me, but somehow I figured it out. So if I did, you could. So thank you very much for your attention, and it's been a pleasure meeting you.
Thank you. We're going to have time for one question.
Please.
We've seen a lot of, I'm not the only person who, I do a lot of endocrinology, but I've seen a lot of patients with new onset type 1 in the last 24 months than 5 years before. The problem is they always end up in the primary care physician and they get metformin. And then they get to us, it's over the 90 days. And I know
Right.
it's frustrating because I know they're going to the study. And there are not many studies right now for these patients anyway, but even if they come to us before the 90 days, what are the what's happening in the arena where you are involved to make the day a bit longer? Because it's really hard for the adult to catch those patients and be phenomenal candidates.
Right. So, so as I mentioned, I think there are trials that are now expanding the treatment window or the randomization window to a longer period of time. Again, the data support that. On the other hand, the counter argument is that you want to intervene as early as possible, right? And the other complication is that after some time from diagnosis, the decline of insulin secretion slows down. And so what that means is that basically you need to have probably more subjects in the study in order to be able to appreciate the difference between the treated and the placebo group. A trick to avoid that is to perhaps do studies that have a more narrow age range, because we know that the younger people have a faster progression, they lose more profoundly. And so many studies have been done within 3 months from diagnosis from 5 to 45 age range. And that to me is so diverse, even though in practical terms, most of the subjects that get randomized are actually young, because the older people, they're less likely to participate in a trial. But nonetheless, if you can actually narrow that down, it allows you to have more power and still maintain a reasonable number of subjects and affordability in the trial. But I can tell you, we had just designed a clinical trial. I was mentioned before, the low-dose ATG and the combination with low-dose IL-2. And that trial is a 4-arm trial, and it enrolls between 8 and 21 years of age for that reason, to keep the decline kind of narrow. And but we still need 124 subjects. And so short of it is a $20 million trial. So, but I think that it's happening, that people are more open-minded towards opening up to longer timeframes for enrollment. And I think patient advocates and patients should make their voice heard, because at the end of the day, it is a chronic disease. Yes, you want to treat as early as you can. Yes, there are all kinds of considerations. But at the end of the day, if we only have a therapy that works, if it's caught too early and all that, it's very limited efficacy, right? So we want to be as effective as we can and benefit the most patients.